All right. Hi, everyone, and welcome. Dr. Matthew Dodd is a geochemist in the School of Earth and Oceans at the University of Western Australia, focusing on the development of a novel geochemical tools to understand how carbon and nutrient cycles affect Earth's long-term habitability. He earned his PhD from the University of Chicago, a University College of London in 2018. From 2019 to 2021, Dr. Dodd was a postdoctoral researcher at the China University of Geosciences in Wuhan, where he developed a novel geochemical proxy to reconstruct ancient ocean phosphate levels. In 2021, he joined the University of Western Australia as a Forrest Fellow investigating how fluctuations in marine phosphorus influence Earth's oxygenation history, climate transitions, and mass extinction events. Currently, Dr. Dodd holds the Australian Research Council Discovery Early Career Researcher Award Fellowship at the University of Western Australia. With that, please take it away, Dr. Dodd.
Okay, thanks, James. All right, so today I will be talking about all things phosphorus, with a specific focus on the Ediacaran period. So to kick things off, um, how important is phosphorus? Well, inorganic phosphorus or phosphate is a major component of all life on Earth. As far as we know, all life forms require phosphorus. It plays a major role in DNA, RNA, so without it, we wouldn't have any life on Earth. Um, and phosphorus only has one major source, which is weathering of rocks. Okay, and that's a big thing. Okay, so when we talk about the major elements or nutrients that life need, we usually talk about things like carbon, hydrogen, nitrogen, oxygen, etc. But most of these elements you can find at Earth's surface in large quantities. Plenty of carbon is CO2. Same goes for hydrogen and oxygen as water. And the atmosphere is chock full of nitrogen. So, out of this critical list of elements that life needs, phosphorus is the only one which tends to be a little bit harder to find. And because of that, a lot of speculation has been made around phosphorus being what has been dubbed the ultimate limiting nutrient for life in the oceans over geological time periods. So, based on that, if we have a hypothetical event that puts a load of phosphorus into our oceans, we can potentially get a bloom of photosynthesis, like this picture in the top left here you see, which is a bloom of algae off the southwest coast of England. But while phosphorus can be a beneficial nutrient for life, it also has adverse effects. So if you have too much phosphorus, then we get the well-known phenomenon, which is eutrophication, where we have excess phosphorus loading, which results in algal blooms or lots and lots of primary production, which then consumes oxygen in the water column as that primary production undergoes respiration and consumes all the oxygen, which is not good for the fish or whatever animals or oxygen-dependent life forms are in that water body. And then the other thing is photosynthesis, as we all know, produces oxygen at the expense of CO2. So therefore, not only can phosphorus affect things like oxygen production via photosynthesis, it can also draw down atmospheric CO2 levels as well. So phosphorus plays a big role in regulating Earth's atmospheric O2 and CO2 concentrations, and in turn, the evolution of oxygen-dependent life forms and climate change over geological time. So I'm going to start by giving a very, very crude overview of the marine phosphorus cycle. So, as I mentioned at the start, all of our phosphorus comes into the oceans from the weathering of the continents. And this is relatively a very small source of phosphorus in the oceans. So if we look at the modern marine phosphorus budget, only 4% of the phosphorus that enters the oceans is coming from these river influxes or from marine sediment recycling. The overwhelming majority of available phosphorus in the oceans is actually coming from internal recycling within the water column. And what I mean by that is organisms take up phosphorus to build their organic tissues. They then die, decompose, and then release that phosphorus back to the water column. But what's really important or has emerged as an important property is the cycling of phosphorus within marine sediments. Now, while it may only comprise 4% of the marine phosphorus cycle, they have a really interesting feedback with redox conditions. So, very crudely, as I said, when we have anoxic waters overlying marine sediments, we find that phosphorus is released from these sediments preferentially. So we tend to see larger fluxes of phosphorus from sediments that are overlain by anoxic waters. Whereas the converse is true when we have marine sediments overlain by oxygenated waters, we actually find that the phosphorus released from the sediments is a lot more muted due to trapping of phosphorus in the sediments. So building on this principle, there was a really great paper published back in 1996 where people looked at how this redox phosphorus control could affect atmospheric O2 levels over geological timescales. So in a hypothetical scenario, if we have a decrease in atmospheric O2 levels, this is going to result in less oxygen in our oceans and a spread of anoxic ocean waters. This in turn increases the amount of phosphorus that's released from sediments that are overlain by anoxic waters. And now we've got lots of excess phosphorus in our ocean, which is driving higher productivity and generating more O2, which counteracts that initial O2 loss from the atmosphere. So this figure on the right here in the bottom right is a great example of this phosphorus redox feedback. So you can see that if we don't have this phosphorus redox feedback, atmospheric O2 levels in this scenario crash and take hundreds of millions of years to recover. Whereas if we have this redox dependent phosphorus feedback, we see that the O2 crash is a lot more restrained, and that's because of this negative feedback cycle that we see here. So this redox dependent phosphorus cycle has been at the sort of at the core of a lot of models in how we understand the evolution of atmospheric O2 over Earth's history. But what we know about oxygen and phosphorus over Earth's history is still very much very hotly debated. So on the right here, we have a couple of figures which show what we believe is the trajectory of atmospheric oxygen over Earth's history. This is fairly well mapped out at this stage. But if we look at the panel below that in panel B, this is a compilation of different estimates of what marine phosphorus levels have been doing over the course of Earth's history. And you can see that the estimates vary considerably, with some saying that marine P levels have been pretty much the same throughout Earth's history, with a few blips here and there. And others saying that for most of Earth's history, throughout most of the Precambrian, marine P levels were incredibly low. So the problem is, the reason we get this spread of values is there are very few tools or geochemical tools at our disposal that we can use to estimate what ocean P levels were actually doing and how they tie into this evolution of atmospheric oxygen over Earth's history. And so the reason for a lot of these differences in opinions of how marine sea levels have changed over Earth's history is that there are a number of competing ideas about what controlled ocean pee availability throughout Earth's history. So, as I mentioned at the start, sedimentary phosphorus recycling, very, very important. Really interesting feedbacks between anoxic and ferruginous or euxinic conditions. But other competing ideas are things like iron oxide scavenging, which was put forward back in the early 2000s. And the idea was pretty simple that in our Precambrian oceans that we presume were fairly ferruginous, we would have had a lot of iron oxide precipitation and formation. And this would have scavenged phosphorus from the oceans and effectively drawn down ocean phosphorus levels. Since then, we've had another idea which was very interesting, published back in 2017. There was some really interesting papers put forward about oxidant-limited organic matter remineralization. And these early papers suggested that under our oxidant-limited Precambrian oceans, phosphorus recycling from organic matter would have been severely inhibited, and that would have been another cause which would have lowered marine phosphorus levels. And then there's been a number of different ideas around weathering, such as supercontinent cycles or orogenesis, which have affected weathering fluxes of phosphorus to the oceans, which have all been put forward. So, long story short, a lot of different ideas floating around as to what controlled phosphorus recycling. So, in this talk, I hope to shed a bit more light on what all these different ideas could mean. So, the Ediacaran period, that's what I said I was going to talk about. And the Ediacaran period, as some of you may know, is really, really interesting, not just because we have some of the first animal life appearing on Earth. We also have these very, very big carbon isotope excursions. And one of the more famous ones, dubbed the Shuram excursion, is highlighted here on this slide in this red interval. Now, the Shuram excursion, while being one of the largest carbon isotope excursions in Earth's history, is very strange because at the minimum of this excursion, the carbon isotope values that we believe reflect seawater DIC carbon isotope compositions goes below the mantle average carbon isotopic composition. So this implies that we must have had a source of extremely light carbon being thrown into the Earth system, Earth surface system to give us this excursion. So aside from it being a really interesting and big excursion, there's another few key points that make this Shuram excursion really interesting in terms of being studied for its phosphorus cycle, but other factors. So the Shuram excursion is believed to represent an oxygenation event, contested by some, but this is one idea. And during this Shuram excursion period, an oxygenation event, we see some evidence for the evolution of early animals. So it's a really interesting time, not from just a carbon cycle perspective, but also from what that was doing to our oxygen cycle and the evolution of oxygen-dependent animals. So, hypotheses for what caused this huge carbon isotope excursion have revolved around the oxidation of organic matter. And that's primarily because, as I said before, we have to get that carbon isotope signal below the mantle value. And organic matter is a great source of light carbon. But But other competing ideas such as diagenetic alteration of carbonate or um, funky carbon dioxide hypotheses have been put forward. When we conducted this study, we thought, can we try to tease apart these hypotheses based on what the phosphorus cycle was doing throughout this excursion? And the reason we thought we could do that with phosphorus was because phosphorus is so intimately linked with the carbon and oxygen cycles that it may very well shed some light on what was happening throughout this really interesting carbon cycle perturbation. So our tool of choice for this study was a relatively new proxy that we developed a few years ago now, which was carbonate-associated phosphate. So this is very similar to carbonate-associated sulfate, which a lot of people will be familiar with. And so the proxy is, at its simplest form, is the incorporation of phosphorus from seawater into carbonate minerals as they precipitate. And so what we found through a number of experiments and various modern analogs was that phosphorus in seawater is often proportionally linked to the amount of phosphorus that is found in carbonates that precipitate from that seawater. And so this gives us a potential proxy that we can use based on the P content of carbonates to estimate what seawater peak concentrations were. So you can see on this graph in the bottom right, we have these linear trends linking P in seawater on the y-axis with carbonate-associated phosphate normalized to the calcium magnesium concentration in carbonate. All right, so we took this proxy, the CAT proxy, and we applied it to a number of different carbonate sections, which preserve the Shuram excursion from all over the world. But this is one of the better studied sections, the Julong section in South China. And so what you see on the far left panel is the carbon isotope composition of the carbonate rocks. And we see we have this big excursion representing the Shuram excursion as values drop from around +5 to around -9 per mil. And then we have our CAP data in the panel next to that, just to the right. No, excuse me. And what you can see in the CAP data is we have this sort of M-shaped pattern where CAP goes up at the start of the excursion, down, up, and down again. And we've color coded the different carbonate minerals based on whether they're calcite or dolomite. And that's because they do affect the CAP concentration, with dolomite typically incorporating more phosphorus than calcite does. And then if we move to the right, we have our uranium isotope profile. So this is uranium from bound within carbonate minerals. And for those of you who don't know, uranium isotopes in carbonates have long been used as a proxy for ocean anoxia, with the more negative values being increasing extents of ocean floor anoxia. So those are really the three panels I want to focus on. The other two to the far right are not so important at this point in time. But what we see here, what's really the key takeaway here, is that we see before the Shuram excursion, extensive ocean anoxia are fairly large, as indicated by the negative values. But during the middle portion of the Shuram excursion, we see that the uranium isotope values become increasingly more positive. suggesting an increase in the oxidation state of the oceans and a decrease in the spread of anoxia. And at this point in time, our cap values actually decrease. And this is consistent with that redox-dependent phosphorus cycling I was talking about at the start. So I'll just dive into more details about that in a minute. But first, I wanted to point out that, as I said, we measured cap in a number of sections across the world. So we have a section from Australia, one from Death Valley in the US, one from Mexico, and then a further two from South China and Northwest China. And what's really interesting is from all of these different sections, we see a consistent pattern in the cap proxy data with this peak at the start of the excursion, a subsequent minimum during the excursion, and then a further second peak towards the end of the excursion. And these trends seem to hold true regardless of what mineralogy, and whether they are calcite or dolomite, that are preserving the trends. So, what does all this geochemical data mean? One way that we tried to go about processing this was to use a simple biogeochemical model. So we used the COPES model for those of you who are familiar with it. So the COPES is a one-box ocean biogeochemical model. It's been around for a while and is quite well understood. So we use this model to build on the work of Shields et al. From, this is a paper published back in 2019. I could be wrong. But this was a really great paper where they put forward an idea where the Shuram excursion could have been driven by an input of sulfate into the oceans, which oxidized a large dissolved organic matter reservoir. And this released a lot of carbon-12 or light carbon into the oceans to give us this carbon isotope excursion. So we built on this model. So we used the framework. So in our biogeochemical model, we have an ocean that is filled with dissolved organic carbon. And what we do is we then simulate a flux of sulfate to the oceans, which then oxidizes this DOC reservoir. And that gives us this carbon isotope excursion. At the same time, when this is going on, we are getting pyrite burial as a result of this sulfate oxidation of organic matter. And this resultant pyrite burial starts to increase atmospheric O2 levels, which is what you see in panel C here. And that's also why we get the corresponding decrease in ocean anoxia as shown in panel D. Now, the real important thing here is the panel B, which is the ocean phosphorus concentration. So what you can see here is at the start of the carbon isotope excursion, we get this large peak in the ocean phosphorus reservoir. And this is coming from a large influx of phosphorus into the oceans as a result of enhanced weathering. The reason we get enhanced weathering at the start of the carbon isotope excursion is because we are releasing a lot of CO2 from that organic matter oxidation. And therefore, this CO2 is feeding into continental weathering and P delivery to the oceans. So that's why we get that first marine phosphorus spike there. We then see this spike decrease to a minimum. And this decrease is attributed to this increase in atmospheric oxygen. So as atmospheric oxygen levels increase, the amount of phosphorus in our sediments begins to increase because more P is being trapped as a result of these elevated oxygen levels in the ocean. And then as we move towards the end of the event or the end of the carbon isotope excursion, we are decreasing the amount of sulfate that's going into the oceans, and we're therefore decreasing the amount of organic matter that's being oxidized by the sulfate. As a result, we have less pyrite burial, and therefore O2 levels start to drop. And for the same reason that the phosphorus levels decreased in the first place, they now increase again to give us that second peak because O2 levels are dropping, and we have less redox-dependent peat burial in our sediments. So what's really interesting about this data is the fact that we see elements of this redox-dependent peat burial happening during the Shuram excursion. But what we see either side of the excursion is really very interesting. So when we are looking at our data before the Shuram excursion, when the uranium isotope data tell us that there was widespread ocean anoxia, our CAP data tell us that marine P levels were relatively low. Same for after the excursion, when the extents of marine anoxia increase at the end of the excursion, we also see the CAP data decline to a minimum. Now, this does not follow that redox-dependent P cycling that I mentioned at the start, because if our ocean is broadly anoxic, we expect our marine P levels to be higher because no longer is P being buried in the sediments under these anoxic conditions. So as a result, what these red boxes delineate are points in time when the marine phosphorus cycle is not following the expected trends or behaviors of this redox-dependent P burial. So let me make that easier to visualize for you. So in the right here, we have our biogeochemical model results. And what I really want to draw your attention to here is the two black arrows. Okay, so we have a number of different model results here. The light blue line is what happens when we don't have redox-dependent phosphorus burial. Okay, so we see that the marine phosphorus level is the same when ocean anoxia is high and when ocean anoxia is low. Okay, but the red line, this is your sort of typical modern ocean P redox-dependent behavior. And you can see that the red line before the Shuram excursion is much higher than the light blue line in the phosphorus reservoir. So there's a big difference there, okay? So we're not seeing that drawdown of P levels when anoxia is high. So what this means is that when there was a large extent of marine anoxia in the Ediacaran period, we weren't seeing correspondingly high ocean phosphorus levels, which we would expect if the ocean was following this redox-dependent phosphorus feedback. So what does this mean in terms of our long-term reconstructions of the phosphorus and oxygen cycles? Well, if this redox-dependent phosphorus recycling is not happening, then what we can expect is that atmospheric O2 levels are not being buffered by this negative feedback cycle. So this would mean that if we have low atmospheric O2 levels, we're no longer getting those high phosphorus concentrations we need to increase those atmospheric O2 levels. And as a result, ocean anoxia could have been prolonged, and therefore we could end up in a situation where we are locked in this low oxygen world because we don't have that P feedback, that phosphorus feedback to push our O2 levels higher and bring us out of this low oxygen world. So a few key points that I'd like to put forward here at the end. So um, our model for the Shuram excursion really nicely fits in with a sulfate oxidation of a DOC reservoir, as it really nicely explains our CAP data, as well as a number of other things like uranium isotopes or redox proxy data, as well as the carbon isotope profiles. Um, Low ocean phosphorus during periods of ocean anoxia imply that the modern phosphorus feedbacks that we see were dampened in the Ediacaran. And as a result, this may have locked the Ediacaran and potentially much of the Precambrian oceans into a low O2 state because we don't have that P feedback to drive our O2 levels higher. And just touching on what's sort of really interesting to me, and I think a lot of others in the community, which is why was the Ediacaran phosphorus cycle different to the modern? Um, what could have maintained phosphorus at these low levels when the oceans were largely anoxic? Um, when did modern phosphorus cycling evolve? That's really interesting. If we want to get a whole understand of um, how did we transition from this low oxygen world to this modern oxygen level world we live in today? And then that leads into a more fundamental question, which is what role did phosphorus actually play in the evolution of atmospheric oxygen? Um, was it a critical factor or was it more of a sideline player? Either way, really interesting points of discussion going forward. So with that, I'd like to wrap up. So I think we've got lots of time for questions, hopefully. So yeah, thanks for listening.
Thank you so much, Dr. Dodd, for a fantastic talk and joining us all the way from Western Australia. We really appreciate you taking the time to be here. If you have a question, please use the raise hand feature or type it into the chat and we'll call on you in turn. And out of respect for Dr. Dodd's time zone, we will be cutting off the questions at the hour. So looks like the first question is Greg, if you want to go ahead.
That was really fascinating and thought-provoking. I enjoyed it very much. Um, I'm wondering though, we have another record of phosphorus utilization in the biosphere, and that is from paleosols, and that shows fairly clearly that Ediacaran usage of phosphorus in soils on a rate basis was an order of magnitude less than modern, and the modern regime started up basically in the Devonian. So if you have an order of magnitude less phosphorus getting into the ocean, how does that affect your modeling?
Hmm. Um, well, that's a great question. Yeah, that's really interesting about the paleosols. If we had less phosphorus going into the oceans, that would doesn't really affect the redox dependent cycling that much, because that's irrespective of how much phosphorus there is in the ocean.
Right.
But what it would do is if you'd have less phosphorus going into the ocean, you'd of course have less primary productivity occurring, and therefore you'd have less oxygen production. So this would just add to the compounding effects that would support this low oxygen world. Um, but it would also mean, I guess, for when I talked about the model earlier, that first big spike we saw in phosphorus, the model suggested that would come from weathering. So potentially that could mean that spike could be a lot smaller if the amount of phosphorus that you can weather based on the paleosol data was less. So that'd be really interesting because we don't have a lot of constraints on weathering fluxes of phosphorus in deep time. So if
Yeah, and most of the paleosalts we have are kind of very arid. There's not a lot of flow through from them into the ocean, so that would compound the problem quite a bit. But the second issue I have is the Shuram. I think the Shuram is an anoxic event because it was like it actually corresponds to one of the big mass extinction events, the one that takes out the ECAP acritarch assemblage. I think it was a high CO2 due to flood basalts in Ukraine. And it's just a replay of the Permian-Triassic boundary crisis, basically. And that's the cause of the anoxia. If that's true, then phosphorus would just ride on that, right?
Yeah, exactly. And that's unfortunately I'm missing the slide for that. But we did explore a number of alternatives to this sulfate oxidation process. And one of the alternatives we looked at was if we had a volcanic event and we have a lot of CO2 release, potentially carrying um,
Like,
uh, light carbon into the oceans. Um, so in this scenario, you'd get enhanced weathering from the larger CO2 levels and therefore more phosphorus input to the ocean.
right.
Um, but because we don't have that pyrite burial occurring, what happens in the model is that the phosphorus concentrations increase at the start of the excursion, and then they plateau and they remain high until the CO2 source is gone, and the weathering ceases. Um, and that's because we don't have the oxygen levels do not rise; they rise gradually over the event, peaking towards the end of the excursion, as a result of this phosphorus input. So the model in that instance could not reproduce the M-shaped pattern that we saw with the CAP data, that up, down, up, down. So that is why we favored the other model, because we couldn't reproduce that CAP trend with the volcanic driver.
Fascinating. That's in your 23 paper, is it?
I think so. I think it should be in the extended supplementary information. Yeah.
Excellent. Well, thanks so much. It's very thought-provoking.
Great question. Next with their hand up is Cole from the basement of Penn State. If you want to go ahead, Cole, go for it.
Wow, good eye. Uh, by the way, Greg, sorry, I don't know your last name. It is in the extended stuff in the back of the Ediacaran paper. Yeah, coming from Penn State, really awesome talk. Loved it. I am curious if you did have this long period of anoxia or anoxic conditions, especially in shallower waters, that would, in theory, be getting some phosphorus through river and inputs. Wouldn't we expect to see phosphorite packages like forming because of the anoxia? And do we see those in like do phosphorite packages occur in like the Duanshan, I don't know if I'm pronouncing that right, carbonate platform in time with that?
Yeah, no, great question. So with phosphorites, the key thing for the formation of phosphorites is you need to trap the phosphorus in the sediments to form them. And to do that, you need to have at least suboxic conditions. So it's very, very rare, or at least I have not heard about or come across phosphorite forming under strictly anoxic conditions, because we are lacking that ability to trap the phosphorus in the sediments, without having some degree of oxygenated conditions. So, in theory, you could have an ocean that's chock full of phosphorus, but never form any phosphorites if it's all anoxic, right? Um, so there's a little bit of an interplay of redox conditions in the formation of phosphorites. But moving on to the other part of the question, do we see phosphorite deposits during the Shuram excursion or the Dushantuo? Yes, we do. Although they are not widespread. They form in specific oceanic regions. So in South China, there is this interpreted, how do I call it, distal shelf bank, which is believed to have been relatively shallow, formed towards the edge of the platform. And there we see a lot of phosphorite formation, and it is commonly linked to these carbon isotope excursions. So as well as the Shuram excursion, there's a lot of other smaller carbon isotope excursions throughout the Ediacaran in South China, and they're very commonly linked with either large or small degrees of phosphorite deposition. So there's definitely something very interesting going on. Whether it's redox or whether it is purely phosphorus fluxes, there's something going on there to affect the formation of these phosphorite deposits at those points in time.
Awesome question. I will ask a question because I don't see any other hands up. I followed your work pretty closely because I work with carbonate-associated phosphate. And one of the things that I've noticed is you've used sort of calcite and aragonite, and you've plotted some great stuff with that. What are your thoughts on early dolomitization, later dolomitization, and how that affects the cap values? I heard you say that some of the dolomites do have higher cap values, and if you could explain that a little further, please.
Yeah. Yeah, so it's a really interesting thing, cap preservation, different carbonates. So generally we find that the dolomite has more phosphorus in them. Why this is, I can speculate and say dolomite may be able to support more trace element incorporation into its crystal structure. What's really interesting is if we look at, so it's not just dolomite, but also if we look at aragonite and calcite. So aragonite also incorporates more phosphorus than calcite does. And again, that may be due to the fact that, I mean, aragonite incorporates a lot of elements more preferentially than calcite does, which again may be due to the more disordered crystal structure of aragonite compared to calcite. So when we've looked at some modern drill cores, and because in the modern ocean, aragonite is generally the primary precipitate, what we see in the drill cores is at the top of the drill cores, we have a larger proportion of aragonite to calcite. But then as we go deeper into the drill cores, we progressively undergo more and more diagenesis, and that metastable aragonite is converted into the more stable calcite form. And as we go down the drill core, and as we see this transition from aragonite to calcite-dominated sediments, we see that the cap value decline exponentially. So it's really interesting to see that that mineralogical control on the cap values. And as I said, I think that is largely due to the crystal disorder and phosphorus being removed during the ordering process. Regarding the part about dolomitization, one of the drill cores we looked at, one of the modern drill cores, did actually have a dolomitized section in it. And what we found was that the cap values shot up really high at the sort of at the boundary between the undolomitized and the dolomitized carbonate, which looked very, very well. It was very obvious that that was altered compared to the part above it. But as we went deeper through that dolomitized section, the cap values dropped back down to what they were above the initial interval. So it really looked like the alteration was just in that initial boundary. And when we dug a little bit deeper into that, we found that it not just the cap values were high, but also the bulk sediment phosphorus were higher at that interval. Um, and also it was also an unconformity as well. So I think what was going on there was we may have had a hiatus in deposition, and therefore this would have been like exposed for a long time, and we could have had phosphatization of that exposed carbonate seafloor, which may explain why the cap values were so high. Um, but interestingly, as I said, the rest of the dolomitized interval, the cap values were consistent with what was going on with the calcite. So whether dolomitization affects, whether post-depositional dolomitization affects the cap values, it's hard to say. I think it would really be dependent on how that dolomitization came about. Um, in the Precambrian, there's a lot of speculation that dolomite may have been, if not a primary precipitate, but actually a very, very early carbonate phase. So in that cases, it may well capture primary seawater chemistry. Whereas if we look at some of the Phanerozoic dolomite, there are instances where it's just very, very clearly it's been post-depositionally altered. And in those cases, it's a little harder to say what's being recorded then.
Thank you very much. Cole, you have another question?
Yeah, I was just curious, if when you were doing any of this CAF work, you ever did any analysis of the carbonates that can be found in marl, in like in those limestone marl sequences. Just if we're talking about, in your talk about the conversion of aragonite to calcite, I'm thinking about like Axel Munnecke's theory of differential diagenesis, and if there happens to be any data about the limestone marl differences in the same unit.
No, that's a great question. We have not looked at that, no. So I wouldn't be able to say. Generally, when we've chosen samples for analysis, we always wanted to choose samples that had as high a carbonate percentage as possible. Um, because yeah, the lower it goes, the more the chances you're going to pick up something that's not your carbonate bound phosphate fraction. Um, I think yeah, it would be really interesting to see what goes on there. But yeah, I couldn't say what would happen with those marl carbonate layers.
Awesome. Thank you.
Awesome. Fred, do you have a question?
Yeah, hi Matt. Thanks very much for a great talk. Um, I'm sorry if I missed this. I may have missed something, but I was wondering whether you could. It's more of a methodological question, and I guess it sort of ties into the question that Jim had on the dolomite issue. But I was wondering whether you could talk a bit about sink switching and how we know that the cap phosphorus is all actually just phosphorus that was incorporated in the carbonate and not just phosphorus that, at least some of it, was sink switched from organic carbon or iron oxide bound phosphorus.
Yeah. Yeah, it's very good. It's a very good point. So that would be a diagenetic phosphorus flux. So as organic matter is being remineralized in the sediment and releasing phosphorus, or similarly, iron being reduced and releasing phosphorus in the sediments and then being incorporated into that carbonate. So that really comes down to is the carbonate preserving water column phosphorus concentration information, or is it recording diagenetic pore water information? And so I guess the ways you could go about that would be looking at whether there's any correlation between the carbon isotope composition of the carbonate and the cap data. So if there was a strong negative correlation, so if you had depleted C13 carbonates with high cap values, that would look a little bit suspect. With regards to switching from reduced from iron to P being incorporated in carbonate, that's a little bit more tricky. I wonder if you could look at iron contents in the carbonate, perhaps. Maybe that might shed some light, see if there's any correlation between the iron content and the P content. That would be some of the ways I might look at that. But really, it's a diagenetic question. So it's what are your carbonates recording? Are they recording pore waters or water column chemistry? So yeah.
Yeah, thanks. I guess I mean, dolomite incorporates iron preferentially, like relative to calcite or aragonite. So if you've got higher iron in the dolomite anyway, would you not extract phosphorus if the iron is also has associated phosphorus before it was incorporated? So you were saying that you had some peaks in phosphorus in the dolomite. Could it not be iron-bound phosphorus originally that has been sync switched to carbonate-bound phosphorus?
I don't know. Could be. But I mean, those peaks that we saw in the dolomite, if I just go back a few slides. So you can see that the peaks are not tied to any sort of one mineral. So if we look at the Australian section, that's pretty much all calcite. And then the Shushang section in South China, that's like a weird mix of calcite and dolomite. But calcite, the cap data starts low and then finishes high in calcite. So I wouldn't say that there is a dominant dolomite control on the cap trends, but they do indeed record a different, incorporate more phosphorus than the calcite counterparts do. So whether they incorporate more diagenetic pore water phosphorus, that's an interesting question. I don't know. What's really interesting is there's a strong correlation that we found between phosphorus contents in carbonate and magnesium. So generally, the higher the magnesium content of the carbonate, the higher the phosphorus content. Why that is, not 100% sure, but again, I was speculating that it might be something to do with the carbonate crystallinity. So you can get some more sort of disordered calcites with more magnesium in them, which may explain why you also get more like rubbish in them, like phosphorus, potentially.
Thank you. Cheers.
Yeah, just continuing on Fred's question, I'm wondering if you could apply maybe sequential leaching like people used for strontium isotope analysis, so where like the first leach you dissolve maybe more diagenetic carbonates, and when middle leach kind of give you sort of more primary carbonate, and residue give you maybe siliciclastic sort of contribution. Could you apply something like this to CAP?
Yeah, and we did indeed. So when we were trying to develop the bulk extraction method, we took a number of samples and sequentially leached them to see where the phosphorus could be coming from. And what we found is that generally, if we didn't add more acid than there was carbonate to react with, the phosphorus concentrations we were getting in the leaches were pretty steady, pretty constant throughout all the leaches. But once we exceeded the amount of acid needed to dissolve all the carbonate in the sample, then we saw phosphorus in the leaches spike, and that we speculate was due to dissolution of phosphate minerals and other phosphorus-containing minerals in the sample. Regarding whether you dissolve preferentially different sorts of carbonates, that's a really good question. If we have a sample, for instance, that's a mixture of calcite and dolomite, we are going to start preferentially dissolving the calcite first and then the dolomite afterwards. And that's just because dolomite is far more resistant to acid attack than the calcite is. So depending on what your diagenetic carbonate is, whether it be the calcite or the dolomite, you could preferentially be extracting diagenetic versus non-diagenetic. So that's a really interesting one. It would be really interesting to have a sample that has this mix of dolomite and calcite, and then yeah, apply this sequential leaching method to see what that gave us. Indeed.
You could also, I guess, combine it with strontium isotope, or do it for samples for which strontium was analyzed. So basically, if you have a meat cut that looks more like sort of seawater composition, it would be interesting to compare it with cup values.
Yeah, no, it definitely would be very interesting. But then I guess we then run into which element records the primary signal? Does the strontium I mean, we can hazard a guess with the strontium, right? If we know what the seawater strontium curve is, then we know what the strontium, the real seawater signature should be. But what's quite interesting, I don't think I have the data here. I'll just check. No, I just have it here. We measured strontium in some of these Shuram excursion samples. And in some samples, the strontium data was very clearly altered. So the strontium data you see in this figure is what most people accept as the seawater strontium signal for the Shuram excursion. But in the Death Valley section, we also measured strontium isotopes, and they were just way off what this South China section was. But yet the CAP data was still the same, and still preserved the same sort of trend. So when it comes to carbonate trace element chemistry, like there's like an order to what gets altered first, or sometimes there is no order if the altering fluid is just preferentially geared towards one element being enriched or depleted. So it's a real complicated one. But yeah, as you say, if it's samples where you know what the strontium isotope composition should be, then yeah, it should be more straightforward.
So another question, and it's probably comes more from Gramschild work, and I guess I should have remembered because he talked maybe a month ago. Could you go over why sulfate flux increased? I guess it's maybe related to long-term oxidative buildup of sulfate reservoir, and more important, why do you think sulfate flux would decrease towards the end of Shuram excursion?
Yeah, good question. So the first part, I think Graham's paper came off the back of some earlier papers from the early 2000s, where people were looking at how the formation of gypsum deposits have modulated the amount of sulfate in the ocean. So if you have some tectonic cycle that creates, like the Mediterranean basin, for example, and you precipitate out loads of gypsum, then all of a sudden your oceans are lacking a lot of sulfate, which can potentially lead to a number of things. But what we're really interested in the Shuram is before the Shuram happened, there may have been some event which precipitated loads of gypsum out. And that gypsum was then subsequently uplifted and eroded, and then put that sulfate back into the ocean where it was originally. So there is this potential disconnect between the amount of oxidants that accumulate at Earth's surface versus what's actually in the ocean, which is really interesting. So I think there was some papers by I can't remember, published in Science, I think they talked about gypsum precipitation, the sulfur cycle. But so the second part of your question was why do we think the sulfate input decreased? I should have explained that better. I mean, it could have been that the sulfate input decreased, or it could have been the other option was we consumed our dissolved organic carbon reservoir. So it's either we ran out of oxidants or we ran out of things to oxidize. So it could have been either. But what's quite interesting is if you look at the CAS concentration data across the Shuram, there is a decrease in the CAS concentration data after the Shuram. So my money would be on the flux of sulfate was consumed or not enough to consume all the organic matter that was available.
Thank you. And maybe just a short one of the early slides, and maybe it was schematic. It showed this Shuram excursion almost overlapping with all Ediacaran assemblages. It probably was a very schematic slide because this one. So probably Shuram excursion should be shorter based on El Neroni ages and so on.
Yeah, I think this figure is probably a bit out of date now. But yeah, still a lot of work needs to be done on determining the length and timing of the Shuram excursion. I would love to see someone get uranium dating of carbonates perfected. That'd be fantastic, wouldn't it? That would solve all our problems.
Yeah. Thank you, Matt. Thanks for giving this talk in such a late hour on your end. And it's end our sort of pre-summer sessions. So you are last speaker. So we take a break and we reconvene in September. So the first talk will be September 25th by Jeff Verwoerd, probably talking about lutetium hafnium systematics, and when we have a full schedule until mid-December. So thanks everyone, and have a enjoy fieldwork or vacation, whatever on your plate, and look forward to see you in September.
Thanks, Andre. Thanks for the invite and the discussion.
Have a great summer.
Yeah. Thanks everyone. Bye.